All right, welcome everybody. You are at Jimby Live. Today you're going to hear an outstanding speaker, Jennifer Surtees and Sandra Small. They're at the University of Buffalo, and they're going to talk to us about their recently published article in the journal Microbiology and Biology Education, talking about a K-12 outreach project. Into the chat, I've put a link to the paper, and you can go read it if you haven't already. And it is open access, like all the papers in Jimby. So feel free to read today's paper and read other papers that have recently been published, especially in our new special issue, Teaching in the Time of Crisis, where we had over 80 articles accepted for that issue. The structure of today, we're going to hear a short talk from our speakers about their paper. And feel free to put all of your questions into the chat. If you wish, you can put your question posed to the entire audience, or if you want to discreetly pass me your question, you can do so by sending a private message to myself, listed as ASM:Rachel Horak. And with that, I'm going to turn it over to our speakers, and we'll come on after their talk and have a conversation with your questions when they are finished.
Well, good afternoon, everyone. Sandra and I are really excited to be here to share our experience and our process in working with elementary school students. So I'm Jennifer Surtees. I'm an associate professor in the Department of Biochemistry at the University of Buffalo. I'm also the co-director of UB's Genome, Environment, and Microbiome Community of Excellence. Dr. Sandra Small is, among other things, the Science Education Manager within GEM, or the Genome, Environment, and Microbiome Community of Excellence. And I'm going to tell you a little bit about GEM because it is important for the genesis of this project. So GEM is an effort, a transdisciplinary effort across the university to integrate research, education, and community engagement. And so we developed this community of excellence focused on the genome and the microbiome, and with a goal of integrating all of these different elements. So GEM is a group of faculty, students, and staff that really works to engage everyone, from children to the general public to other academics, to improve microbiome and genomic literacy and to foster knowledge in many, many different ways. And so with this particular project, we were really interested in introducing the idea of the microbiome to elementary school students, and to sort of change the way they think about microbes, from being kind of icky and bad to actually being really important and typically good. So our goal was to develop a multidisciplinary approach that engages all students, those who are interested in science and maybe those who are maybe not so much. We really wanted to integrate microbiome literacy into the second grade curriculum. So it was a natural fit through literary practice, social studies, and the arts. We wanted hands-on inquiry-based experiences, and we wanted them to be able to relate the microbiome, what they were learning, to their own body, and to engage their imagination through art, movement, and storytelling. So our message, as I said earlier, is that microbes are all around us, in us, and on us, and that's a good thing, not an icky thing. And to integrate into the second grade curriculum, and I have to say, my daughter was in second grade at the time, and so I had a sense of what was going on. So when I went with my daughter on field trips of different types of communities, and so communities are made up, they learn that communities are made up of different people and different homes and different businesses with people with different jobs. And so this was a perfect fit for the microbiome because they live in communities on our different parts of our bodies and with lots of different jobs. And just as the world around us is made up of different habitats where different plants and animals can grow and thrive, our bodies have different habitats for microbes, with different microbes preferring different parts of our bodies. So we are a series of habitats. So to convey all of this information and to develop partnerships with our elementary school, our local elementary schools, we developed this 3-day progression of a workshop so that we could actually go back into the classroom multiple times to develop relationships and engage with the students and with the teachers over time. So just very generally, on day one, it was really building the core ideas of what the microbiome is and what its the jobs of these microbes are. Day 2 was to enact the scientific processes, and day 3 was creating models. And there's typically about a week between each of these sessions. And we'll spend a little bit of time going through in detail what we actually did and why we did it. So on day 1, we go into the classrooms and introduce the microbiome in general. So there are key concepts that we want to get across, that microbes are tiny and we need a microscope to see them. Habitats and so that the microbes live in different parts of our body. They live in communities, they have jobs, and their diversity is really important. So in order to really kind of assess how they are thinking about microbes and if they're thinking about microbes, we start with a KWL, or want to know and learn what we learned chart, to assess their prior knowledge of microbes. And we'll show you an example of that in a little bit. And then we found this great book, which we have no association with whatsoever, called Tiny Creatures: The World of Microbes. And it's a phenomenal picture book for this age group, or just this age range, to increase the conceptual knowledge, to develop literacy and vocabulary, and to learn more about the microbes. So this is our KWL chart. And this is the book, which I highly, highly, highly recommend. And this is me reading this to a group of children, and us talking about different types of microbes. And I should say, this is one of our graduate students. And graduate students, we engaged a lot of graduate students, and have over the years engaged a lot of graduate students and undergraduates, training them so that they can go into the lab and work with the students as well, which is a great opportunity and experience for the graduate students. So this is an example of a kind of a filled in KWL chart, where what they knew to start with, there wasn't a lot in there, that they're small, that microbes make you sick, that you need a microscope, and then lots more of what they want to know. Lots of really interesting questions about what microbes look like. Do they have ears? How do they eat? And that sort of thing. And then after talking as a group and reading the book and doing some explorative discussion, they learn a lot from us, from the book, and from each other. And so they come away from this very first session having a broadened conceptual knowledge of the microbiome, and microbes in general, which is really fun to watch. They don't all still love the idea of microbes, but they have a different way of thinking about it. So to build on these ideas, we have them look at baker's yeast through a microscope. And we use baker's yeast primarily because I work on yeast in my own research lab. And so we have ready access to it, but you can also obviously buy it in the grocery store. And then we had them collect microbial samples from their classroom habitats, including sometimes from themselves. And this is to relate the idea of microbes being everywhere, including in their own environment and on their own bodies. So they use sterile swabs, and they take samples from themselves or from their environment. And then they streak those cells that they sampled onto a plate of rich media where a lot of different things will grow. Then all of these plates are parafilmed, and they do not get opened up after that. They stay sealed for the remainder of this exercise. We leave the plates in a warm part of the classroom to grow. The cells then grow, and they divide, and they form colonies that are visible. And these different types of cells form colonies that look different. And different samples taken from different parts of their body or different parts of their classroom look different as well. So we just have a few pictures in here of students labeling their plates and streaking out their samples onto their labeled plates, and then looking through the microscopes at some yeast samples. And I think we also have some stained bacterial slides as well for some of these things. So I'm going to let Sandra take over, and she can talk a little bit about day 2. So we go away for a week usually, and then we come back and they actually observe their microbiome and fill out worksheets and use both observation, writing, and drawing skills. So Sandra?
Thank you. Yeah, so day 2 is a lot of fun because up until this point, there's been a lot of talk of conceptualization and what do you think it's going to look like? And the students have a lot of anticipation. One of the first things they notice when we start is the smell. So we emphasize to them that they're going to use their sense of sight and their sense of smell only to do these observations. And then we review the KWL, what did you know, what did you want to know? And ask them, what did you expect? What do you think your microbes are going to look like? Do you think from the 2 habitats they're going to be the same? Are they going to be different? And then we give them back their plates, and they get to look at them and compare using a guided. Is that the next slide, Jennifer?
Sorry. Yes.
Okay.
Oh, that's looking at the plates.
So here, just stay here for a minute. So you can see on these, we had them split a plate in half. If we have enough plates, sometimes we give them 1 plate per location. And then so in the middle, Isaac compared his arm and his head. So here we ask questions like, how many microbes do you see from each location? What color are they? What size are they? And draw some of them. So you can see that he had a very good comparison to that they were very different microbes that grew. All right, there we go. So here I am, explaining this worksheet or guided lab notes to these students. You can flip through. So this is our first iteration of the lab notes. You can see we used scientific terms for the students to make their comparisons, draw some of their colonies. We provided a box and just asked them to describe them. And then we talked to the teachers, and we got some feedback from the teachers on that maybe our lab worksheet was a little bit too high level for our intended audience. So we made some modifications to our lab notes. So now you see, this is what we use currently. We did not put the words in for the shapes of the colonies. We merely just put the shapes, and the students can draw what they see. And we added a word bank, because just telling a second grader, draw what you see, that's very nebulous to them, and they're not really sure what direction to go in. So we gave them some words to help them know what they should be looking for. And we used second grade terminology, right? Goopy, fuzzy, cloudy, words that they understand. So we met them where they are, which is what we should have done in the first place, but this is why we're learning. We also, instead of just asking them to describe what they see, we gave them a structured chart. So charting is something, at least in New York State, that most students learn in first grade. So we were reaffirming what they had learned. So here it's guiding them, right? Write down where your sample was taken, and then count the number of colonies. I always tell them if it's over 20, you can write too many to count, because you get some that are just full of colonies. And then draw the shapes that you see in each habitat, or from each habitat. What colors do you see and what textures? And again, reiterating, you're not touching to feel the texture. Does it look fluffy? Does it look goopy? And so they can fill in this chart, and then they can use the chart. Here's the one filled in, so you can see the right hand versus the doorknob. So this student was comparing, because we do get them thinking a little bit about like what habitats come in contact with other ones and might share microbes. So here you're using your hand to touch the doorknob. So maybe the student was proposing there would be some similarities in the colonies that were seen. So she filled out this chart with all the colors and the textures, and then she can take her completed chart and answer questions. Next slide, Jennifer. Yeah. So we'll get to the questions in a second. So here we asked them, this again was one of this was our second iteration of the lab observations. So you can see, instead of the box that we had, we put a circle. So now it looks like the plate, and asked them, which habitat did you swab? And what they were supposed to do, and most students did, was write the habitat name on the line. So this student should have written hand on the line, and then in the circle, draw the colonies they saw. But we realized that we were not explicit in our directions, so we had to make a version 3 of our lab notes where we actually asked them, I think is that the next slide, Jennifer?
Oh, yeah.
Okay.
Sorry. There we go.
There we go. So you can see we changed it, and now we're more explicit with our directions. Which habitat did you swab to obtain this microbiota, and then a line for them to write it, and then separate direction, draw some of your microbial colonies in the circle. So you can see here, I think this is Will, a student, Will's paper. And so Will swabbed a quarter and his right hand, and so he filled in that habitat and then drew what he saw on his plate. So here's where they take the information from their drawings and from their chart that they filled out to answer guided questions. So instead of just saying, are the colonies the same and or different, now we're guiding them through. So are they the same, right? That was our big question when we started this. So they can draw their conclusion, no, they're not the same. We haven't had any students to date have ones that look exactly the same. There's always a difference in color, size, or quantity. And then we explicitly say, what are 2 differences, and give them the lines to write, and what are 2 similarities. We really try to stress for them to write in full sentences, because I don't know how many of you work with kids of any age, but they don't like to write in full sentences. They like to give short answers. And so we're really trying to drive home those literacy skills as well. So here we have students filling out their lab observations, their lab notes, looking at their plates, making their observations. We talked to them about how scientists need pictures as well. You need to write things very explicitly so that you can share that with your colleagues and they can understand, but how pictures and drawings, speak 1,000 words. So that's why their pictures are so important.
So then in the third day, so we, after about usually another week later, so we go through and they write their reports, and then we take the plates away and we autoclave them so that before they get disposed, so there are no safety issues within the school. And then about a week later, we go back and we review the same concepts that we talked about. We remind them of what we learned, their KWL chart, what we learned. Sometimes we reread the picture book, depending on the circumstance. And then we go over these concepts of the diversity of microbes. They come in many shapes, many different sizes. They live in lots of different places, and they all work together as a community and help us and our environment. And we show them these beautiful images of microbes as inspiration for an art project. And we ask them to create their own microbes. We give them art supplies and paper and glue and glitter glue and lots of googly eyes, and they create. They take inspiration from these images and they use their imagination, and they think about. We ask them to, as they are creating their microbes, think about, what does this microbe do? This is one of our graduate students making a microbe himself. And I have to say, I was talking about giving this talk yesterday, and Caleb, this is Caleb, he said that doing these workshops was the most fun that he had in graduate school. He's a postdoc now, and this was the most fun that he had in graduate school. But you can see that some of the students looked at these pictures and did very kind of literal, realistic images, microbial images that look very much like the pictures we gave. And others were much more creative and free with the googly eyes and the pom-poms and the feathers. And it was, anything was wonderful. It was tremendous amount of fun for them and for us to kind of look and see the kinds of ideas they had about how the microbes function, how they work together, how they might look, how they might interact with one another. And then in many classes, we've tried to extend just the art project into storytelling to sort of formalize that storytelling with the students actually writing a story about their different microbes. Sandra, do you want to talk a little bit about these?
Sure. This is probably my favorite part of the whole lesson, is just seeing their wheels turn in their creativity. What we don't have pictured here is my favorite microbe that a student created. And when I asked that student what the microbe's job was, he told me that its job was to go to school for him when he didn't want to go. So I said, well, I wish I could create a microbe for me some days at work when I just want to stay in bed a little bit longer. So here we have some situations. You see Patricia on the right on the blue paper. So Patricia, her job is she's a good microbe, and so she breaks down food. And then you have Strong Sergeant Army Man, and Strong Sergeant Army Man fights the bad microbe. So you can see here that these students were really listening to what we were saying throughout this whole process, and remembered that when it came time to make their own microbes and give them a job.
And it's really important as throughout the workshop on all 3 days to kind of go through as the students are doing things, as they're swabbing, as they're creating, as they're drawing and illustrating, to have these informal conversations with them and encourage them to actually talk to each other with productive conversation, because it really enhances the learning and the conceptualization of everything that we're trying to get across. So finally, we try to put this into, we've talked about these communities of microbes, and we've got our plates, which are our class microbiome of actual microbes. And so we then also put together our artistic class microbiome for each of these classrooms at the end of the session. We'll often take pictures of the students along with their microbes, but we're not showing those images today. Just a little bit of detail. We've done these, or just an extension, excuse me. We've done these this lesson in a number of different environments, primarily in 1st to 3rd grade, but we have modified it for 611 and 1211 classrooms, bilingual and multilingual learners, and we've modified it for middle school and high school, basically asking more sophisticated questions and more sophisticated hypothesis development. And we've also adapted it for family STEAM nights and family STEM nights, taking different elements of it. And it's always a lot of fun, and people learn a lot. Sandra, do you want to talk a little bit about the evidence of learning?
Sure. So evidence of learning, you saw the KWL chart, the microbiota lab notes, the art model, and then anecdotal records. So these are sort of ongoing assessing as we're going through the lesson. We can pivot if we need to go into something a little bit more, if we see a misconception come up. Students' prior knowledge. This is most interesting. Students know micro, and they know that it means small. Microorganisms or microbes is usually a new word to most of them. We've been lucky that in each class, we can usually get one student with some prompting to tell us that microscopes are used to look at small things. A lot of times when I say the word microscopes, the students will say, oh, that's what we use to look at the stars. So then we go in and talk about the different, micro in scope and how those different parts of the word mean different things. And then student observations, like Jennifer was saying before, it's very important to keep conversations going while the students are working on these. It's very easy for them to get excited about the process and sort of forget a little bit about the focus. So keeping on top of that conversation, asking them guiding questions and hearing their answers, because that can help understand, like I had said, how much is actually being retained, what is being retained, are there any misconceptions coming through? Here we talked about going to school when I don't want to, that microbe. So it's just fun to talk as well and just hear what these students have to say in a totally different perspective than we as adults who work in this field think about things.
I should say also that in some of the cases, especially early on, we had in the third session, we had graduate students act as note takers to sort of record the conversations that we were having. And it was clear that over this 3-week period, the students had retained knowledge, were able to answer questions, and were actually able to apply that knowledge in different ways.
And then if I can just follow up on that, we had a group of students, so separately, we had an event about the microbiome with the Department of Dance and a lesson at the Science Museum here in Buffalo. And we brought students who had done this activity 2 years prior to coming to the museum. And when I introduced them and asked them about microbes and what they knew, they were still able to tell me things that they had learned 2 years prior. And so it showed that this was actually working and they were retaining the information in the way that we intended for them to retain it.
So I just want to reiterate our safety concerns. Because we're taking environmental samples, we don't know what they're going to be swabbing. And so we do ensure that the plates are sealed with parafilm. The students and the teachers don't ever open the plates. They observe, as Sandra said, with their eyes only. And the teachers will collect the plates. And we, as a university partner, we typically pick them up and take them away and dispose of them here at the university, where they can be easily autoclaved. This can be done in partnership with a university or other places that industry or company that would have the capacity to do the autoclaving, the sterilizing appropriately, and to help facilitate plates and that sort of thing as well. This is another class microbiome, and this is a whiteboard, or a smart board rather, illustrating some of the things that the students learned, that they talked about having learned after this, and looking forward to learning more. And so this is a terrific experience. And we've been in, Sandra, you're going to have to remind me, but we've been in over a dozen schools. We've reached, I think at this point, probably over 500 students in Erie County and Niagara County and in our general region. And it's really a lot of fun. It's been extremely rewarding to do this kind of work. It's very different than the bench work that I typically do. But it's really, I have enjoyed it so much, and I'm so glad that we have done this, and we're continuing to do this with other topics and age groups as well.
And we have lots of questions from our audience because people were really interested in this work. So first off, I was wondering, tell us a little bit how you got interested in moving from bench science and bringing in lessons into the K-12 classroom. What motivated you to do it, and how did you learn the skills to get involved?
Well, for me, a big part of the motivation was really a really strong interest in promoting scientific literacy in our area, and particularly with respect to the microbiome. And I initially piloted this in my daughter's second grade classroom. So I was really lucky to have a teacher that was willing to allow myself and a graduate student to come in and try this out. And it worked pretty well. And we actually got invited into a number of other classrooms, and the whole workshop evolved. I mean, the structure is basically the same as it was at the beginning, but we've evolved in many different ways. And so Sandra, in addition to having a PhD from the Department of Biochemistry here in Buffalo, is also a certified high school science teacher. And so she has those skills. And so it was a great science education manager for us. For me. And in addition, we also leveraged the Graduate School of Education at the University of Buffalo, including Lynn Shanahan, who was extremely interested in integrating literacy practices into science and social studies to try to increase the amount of science and social studies we're teaching our students.
Thank you. One person asked, what are some suggestions for adapting parts of this activity or the whole activity for online learning? Is it possible?
anything's possible. Do you want me to answer, Jennifer, or do you want to?
Go ahead. I will jump in.
Okay. So this one, because of the safety issues, we don't want to be sending the plates home with the students, but it's something where the teacher or a graduate student in a lab. So I have some connections right now where I have graduate students who are working virtually with some of our local schools, and they can do some of the hands-on things and be the hands of the students because they're not there. So you could have somebody there and the students suggest the locations that you want to sample, and then the person who's doing it, either the teacher or visiting scientist, can do those samples, and then you can talk about it as a group and show the pictures. You don't get that smell aspect virtually, but I think everybody's okay with that. So yeah, I think it could definitely be adapted for virtual.
And you could clearly send art supplies if to different families if that was something that that wasn't available to people in general. But the safety issue is something that we have to be aware of.
You're absolutely correct. So in the paper, you do note about the biosafety guidelines, the ASM recommended guidelines for biosafety, and it's super important that we follow those for all audiences, and we follow your advice. So all the papers that are published in GIMBY that include any kind of bench research are approved by a biosafety committee. So in case you weren't aware. How many schools have you visited using this activity, and how do you usually select the schools that you go to?
So how many schools is it now, Sandra?
I think we're up near 20. Some of those, so I say that, but we go back to a lot of the schools because once the teacher has us in the classroom, we're hearing from that teacher the next year, hey, can we do that again? So 20, I mean, I think it's an impressive number, but if it sounds small to you, just think that some of those we've been to 3 or 4 times.
Yeah, and initially, as I said, was lucky enough to have my daughter's school teacher allow us in, and of course the school allow us in. After that, we've had a lot of word of mouth spread to different schools. So we work with the Buffalo Public School System a lot. We have good connections with them. We also have good connections with Sweet Home School District, which is nearby. But we also have had graduate students who volunteered say, can I bring this to my niece's school and my nephew's school? And we've had faculty members saying, can I bring this to my kid's school? And we are more than happy to have people to provide the plates and the supplies and send people out to all different kinds of schools. And so in that way, it's kind of this natural spread of the unit.
Great. You talked a little bit about adapting your activities, but one participant was especially interested in deaf or hard of hearing students or students with special needs. How might you adapt some of these activities for those students?
So we have not worked with any deaf students to date. It reminded me that we do have a school for the deaf nearby that we should probably reach out. Yeah. I've worked with them in other activities. They've come and they bring a translator who can sign for us and keep up. So I think that this is definitely something that those students can partake in. And then as it relates to the special needs students, so we've done this in 611 and 1211 classrooms. So in those classrooms, we talk with the teachers to understand what the capabilities are of the students. And for some of them, we had the teacher do the swabbing and demonstrate to the class, and then the class talks about it. Others, we helped them swab their heads and then do their own plate. And then other ones we've had where we tell them, this is, we talk about as a class, what community or what habitats would you like to swab? Do you think we should swab? And then the entire class does the same 2 habitats. And then when we're going through the notes, we can do it the same for everybody, and we can fill out the chart and, it's the same habitat. So it's much more guided.
You mentioned 611 and 1211. I haven't heard the term before. Can you explain what that is? I'm I'm only familiar with higher ed, and maybe that's a term used for K to 12 students.
It is definitely a term for K to 12. So they are self-contained classrooms. So they're students who stay with the same teacher and the same assistant all day for all of their classes. So a 611 is 6 students, 1 teacher, 1 assistant, and 1211 is 12 students, 1 teacher, and 1 assistant.
Thanks for that clarification. And another talking about adaptation, how might you adapt the activities for family nights? And maybe even fairs, or when we are back to in-person events, maybe some of these big in-person activities that attract, hundreds of students.
So we've done a few things, and Sandra can jump in as well. But one of the things that we do, because it's a night, we don't have time for things to grow up, is we have volunteers who will volunteer to give their own microbiome samples. So they'll actually pre-swab plates and just push their hand up, or I've had one of my former graduate students would be in the lab, taking her shoes off and swabbing her toes and preparing the plate so that it would grow up in time for the night. And then we bring art supplies so people can create their own microbes and bring those home. Sandra has done some really cool microbe art with people as well. If you want to mention that, Sandra.
Yeah, so we've had some events. We have an open house on our medical campus every year, well, normally. And so as part of that, we do yeast bio art. So we have plates available for people, and then just baker's yeast, and they can use a swab to dip it in and use the plate as their palette. So they get to draw some sort of design, and then they can take that home with them because it is a non-pathogen yeast that we use, and they can take it home and they can watch their yeast drawing develop. So we've had a lot of positive feedback from that. And it's just, again, that's talking less about the microbiome and just making people aware of the microbes that are around us and how we use them for good in our life and deliciousness in our life.
And of course, Saccharomyces cerevisiae comes in pink and white varieties. And so we can actually have multicolored images as a result of that.
Yeah, images and hands-on activities are just so powerful for those younger people. That's great. Yeah. What about did you ever have students ask you about microbes where you weren't quite sure of the answer? And what kinds of strategies would you use to handle these harder questions? Maybe the concept was very complex, especially for the age group you were talking about, or maybe the answer wasn't known, or maybe the answer was outside of your expertise. Did that ever happen to you when you were doing these events, and how did you handle it?
So yes, that happens, and it's awesome when it happens. And I mean, I personally, and Sandra can speak for herself, but I personally handle it the same way I would handle a question I didn't know the answer to at the end of a seminar, so a research seminar that I'm giving. So, if it's a tricky question, it's something that we can try to work through together, sort of make some predictions. So, I don't know the answer to that, and I'm not sure if anybody does. So let's think about what's possible. What could be the answer to this? And sort of approach it that way. If it's something I really don't know, I'll usually go and look it up, and then we can talk about it the next time as well. So, and I think that's a really good illustration of, just because I'm a scientist doesn't mean I know everything, and we can all learn and research and do that kind of thing. I don't know if you have anything to add, Sandra.
I was just going to say, it's okay to say, I don't know that. And it's good. I learned this in my education training, and it was the moment that I think made me a better teacher is when my supervising teacher said, it's okay to say, I don't know, and I'll get back to you on that. So because we go in 3 times to see these kids and we have email communication with their teachers, I'll say, that is a question that I don't know the answer to, and I'm going to look it up, and you can look it up too, and then we can talk about it when we come back together. Or if it's on the last day, I'll email your teacher and we can, I'll get you the answer that way. So it's okay to say, I don't know. And it's really good for them to see a scientist, because I have a friend whose daughter is 7, and she comes to me with every question, and she says, well, you're a scientist, so right? And I have to tell her I don't know everything, and my expertise is in this area, and so we have conversations about how to get those answers.
Another question that came through, what kind of benefits do you get as being scientists and educators and publishing with JIMBY, and what motivated you to write up this curriculum article for JIMBY?
So I think, as I said before, I think scientific literacy is really important. And one of the things that we want to do is increase the number of people and the types of people who are interested in becoming scientists. And so that was a big part of the motivation of doing this project in the first place. I think that I have found, and the graduate students and undergraduates and other faculty who have done this have found it extremely rewarding to kind of to be able to interact and be in a position where you're having different kinds of conversations and different kinds of interactions. And so in that way, it's really beneficial. It makes you think about your science differently as well, how you approach the world of science. And so, for improving diversity, having sort of a fulfilling experience yourself, and then also changing your view, your outlook and your perspective a little bit, I think are all of the benefits of doing this sort of thing. And then I think it's really important to share what we're doing. I mean, to make it openly available and accessible to other people, and share our experiences to encourage other people who are not necessarily trained education researchers, but people who are really interested in engaging elementary and high school students in these different ways, that we can do this, and that yes, you need partners, but it's totally doable, and it's absolutely worth doing.
And I would just add, teachers are very, very busy. We all know that. So if we can help them and give them creative ways to infuse science into their existing social studies lessons, English lessons, that is just a benefit to everybody, to help those teachers have those tools to do that.
So at this point, we have a couple of more questions to get to. I'm going to quickly ask our audience members to take a quick poll to give us feedback on this event today. This event is being made available by the American Society for Microbiology and JIMBY, and we would greatly appreciate some feedback before our webinar ends today. So just take a moment to give us some feedback. Okay, so a couple more questions for our author team. You do talk a lot about how doing this kind of work is important to find good collaborators. And do you ever intend to launch this as a nationwide effort, or would you like to find collaborators outside of the country to work with you in further developing your project or furthering developing your network?
Yes. We would be happy to do that and work with other people. In fact, my sister is, so I was happy to see some fellow Canadians in the audience today. My sister is a high school teacher in Toronto, and before the pandemic hit, we were actually talking about a way of adapting this so that her high school students could go into elementary schools and sort of do this kind of thing in Toronto. And so, yeah, so I think it would be great we are more than interested in broadening our network and increasing this unit across the country and beyond.
And let's think a lot of our audience teaches undergraduates. How could you recommend using this model as a module for getting undergraduates involved with K-12 education and maybe a community learning and community service?
So we actually collaborated, and I'll let Sandra talk a little bit about this. We have collaborated with the Honors College here at UB. they have a first year service learning course that they're required to take. And we became one of their community service partners in doing this. And so Sandra can talk a little bit about that.
Yeah, so right now the Honors College, like Jennifer said, requires students to take a service course. There is a big push, not just at the University at Buffalo, but in the whole SUNY, that's the State University of New York system wide, where we really want to give students more experiential learning opportunities. So this is a way where we can partner. At our university, we have the College of Arts and Sciences, the School of Pharmacy, the School of Medicine and Biomedical Sciences, as well as the Graduate School of Education. So we can bring those science-minded students, undergrad or graduate, together with the education students and get them out there together in teams to go and work with these schools. And that's something that we're doing, and we're working on formalizing it, working on creating a course that students could take for credit. So that's something that we hope to establish in the future. So this is definitely something that's very easy to give to undergrads in that capacity.
And of course, I mean, this is just one unit. There are other units that we have developed and are in development as well that could follow that same model for 5th and 8th grade students.
So you've talked about the students that you worked with, 2nd grade, 5th to 8th. What do you think about going any younger than 2nd grade that you implemented it? Would you think that that would be likely or highly unlikely or not recommended?
I say I've done it. My mom teaches pre-K, so I go into her class. And then I have a friend who teaches kindergarten in Maryland, so I go into her class usually every year. And we just kind of modify the messaging. Yeah. So we don't really talk about so much the communities and habitats, more about just this invisible world that you need a microscope to see. And we let them touch a plate just with their hands to show that we have the special food on the plate that's going to make these microbes grow, and just introduce them to the terminology more so than all of the greater concepts.
Yeah. Yeah, I've done kindergarten as well, and it's fun. You just have, as Sandra said, you have to change your message a little bit, but it's fun.
As a mom of 3 preschoolers, I can imagine that they would love this activity, but we'd have to alter the activity a little bit because they're not able to write yet. But they could.
Yeah, it's all in the kindergarten class. It's all conversation based.
Yeah.
Mm-hmm.
They love looking through the microscope.
And let's see, any other questions, put them in the chat box. We have a question about the bibliography. So somebody asked, the resources are listed in the published paper. And then the book that you indicated, Tiny Creatures, you said that that's the best book that you found to teach the concepts. Have you found any other books that you would recommend for this audience, or any other resources so people can learn more about doing these types of activities?
I read Ada Twist, Scientist. I am drawing a blank on the author. I know it's right around the corner, but I'm not going to go get it. Ada Twist, Scientist is an excellent book to talk about the scientific process and thinking. So that's one that I read with K through 2. You get above 2nd grade and it's a little bit young for them, but it's really great for K through 2.
Yeah, I can't think of anything honestly off the top of my head. I was just going to say in the paper we actually we did include instructions on how to make, in addition to the description of what we did, but we've got instructions for how to make plates if you need to make them yourself. We've got where you can get different kinds of videos if you don't have microscopes to show the different microbes. So we tried to put in a lot of resources. We also put in a table aligning everything we're doing with next generation science standards and Common Core, so that it's useful for teachers.
Go into the chat. If you missed it, there's a couple of great other books that people have used to teach these concepts. I loved Ada Twist. That was a great book. And then ASM Press put together, Invisible ABC.
Oh, yeah. Yeah. I've seen
think
that one.
that's
I have
very
not
fun.
read Do Not Lick This Book, though, so I'm going to have to check that one out.
So that concludes our session today. Thank you very much to our speakers. Lots of positive feedback from the audience. We really appreciate having you here today. We'll have our next event in 4 weeks, and at the same time, using the same Zoom link. And take a look at your registration. We'll have another author of a recent GIMBY article, and you'll receive a follow-up email next week that contains the link to watch this webinar again if you choose. It'll be posted on GIMBY's YouTube playlist. Okay, so thank you for everyone attending, and we'll see you at our next event. Appreciate it.
Thank you so much.
Thank you.